Refrigerator, -20, used to store a variety of samples at a very specific temperature range. Ultra-low temperature freezer, -80, used to store samples for long period of time. Distillation unit, used for remove impurities from water by converting water into steam. Liquid nitrogen containers, are best for storage and transportation. Electronic balance, used for obtaining the weight or mass of objects. Laminar Flow Cabinet, used for safely working with biosafety level. It provides personal, environment and work protection as contamination-free work environment. pH meter, used to measures the hydrogen ion activity in water-based solutions, indicating its acidity or alkalinity, which is expressed as pH. Water bath, used to incubate samples in water at a constant temperature over a long period of time. Hot plate, used to stir and heat the solution simultaneously. Microwave, used for heating samples and solutions for diverse experiments. Incubator, used in tissue culture and microbial genetic laboratories purposes. Autoclave, uses steam under pressure to sterilize laboratories apparatus. Hot air oven, used dry heat to sterilize laboratories apparatus. Mechanical shaker, used to mix, blend, or agitate substances in a bottle or flask by shaking them. Vortex, used for mixing or agitate laboratory samples in test tube for homogenization with high degree of precision. Centrifuge, is a device uses centrifugal force to separate various components of a fluid on the basis of densities at controlled temperature. Light Microscope, a device used to view objects which are too small to see or explore with your eye alone. Mixer mill, grinding small amounts of samples. It can mix and homogenize powders and suspensions in seconds. Also, it disruption of biological cells as well as for DNA, RNA and protein extraction. Spectrophotometer, measures the light intensity as a function of wavelength, and used to measure the concentration of compound in an aqueous solution. Nanodrop, is a quickly and easily quantify and assess concentration of samples containing protein and nucleic acid even with a very small quantity of sample, 2 microliters. PCR, used for amplifying DNA and RNA due to PCR reaction. Real-Time PCR system, is a platform for the detection and quantification of nucleic acid sequences that will not comprise the data quality using specific software, to measure the cycle-by-cycle accumulation of PCR products in a single tube and homogeneous reaction. Agarose Gel Electrophoresis, used to separate and analyze macromolecules like DNA and RNA on the basis of molecular size and charge. SDS PAGE Electrophoresis, used to separate and analyze macromolecule like protein on the basis of molecular size. UV Illuminator, used to view nucleic acid and protein suspended, within, polyacrylamide or agarose gels. Gel Documentation System, offers high image quality and data acquisition for documenting and analyzing electrophoresis gels.